get going. It's been a couple of minutes. So welcome everyone again to the next edition of the 2023 State of Play series. And for those of you on Zoom, you'll probably have seen a notification about the meeting being recorded. So please just click OK or agree to continue. First then, I'd like just an acknowledgement of country. So MND Australia acknowledges the traditional custodians of the country throughout Australia and their connections to land, sea and community. We pay our respects to the elders past, present and future, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am. I pay my respect to elders past, present and emerging, and extend a welcome to any Aboriginal and Torres Strait Islander peoples on this call today. So tonight we'll hear about some earlier stage research looking at ways we can develop new treatments and delivering those treatments. We'll hear from Dr. Margreet and Professor Trent Woodruff, both from the University of Queensland. As with our previous editions, each presenter will talk for about 20 minutes, and then we'll have a combined Q&A at the end of the session. So please submit your questions through the chat or the Q&A function if you're on Zoom, or through the comments if you're watching through Facebook. You don't need to wait to the end, just fire them through and we'll collate all the questions and get them all ready, and I'll ask them at the end of the talks. So you don't need to try and remember your question. So first up, I think Margreet's going to start. So please take it away, Margreet.
Welcome, everyone. It's good to be here. My name is Margreet. I'm coming to you from the Queensland Brain Institute. First, I'd like to thank the organizer for giving me this opportunity to talk to you about new approaches to developing treatment for MND. So the title of my presentation today is Targeting Hyperexcitability in MND. So I will take you through what hyperexcitability is, how it relates to MND, and how we hope to tackle this. So I'm a research fellow at the Queensland Brain Institute. This is a nice image. So the Queensland Brain Institute is in UQ on the St. Lucia campus, and it's home to about 250 researchers. And it really is quite a nice place to try and unlock the mysteries of the brain. So I'm part of the synaptic plasticity group led by Professor Pankaj Shah. And our focus is really the electrically active cells in the brain that we call neurons. So what we can do in the lab is we can record their electrical activity, but also manipulate their electrical activity to try and study their function both in health and disease. Below here is an example of one of these recordings. When a neuron is activated, excited, it can fire action potentials. And this is its way of communicating signals to either other neurons, or in some cases for lower motor neurons, their signal onto muscles to make them contract. So action potentials are generated and sent down the axons towards synaptic terminals, where they move on to either innervate other neurons or muscles. There's not a thought or an action you've had that doesn't involve some sort of neuronal communication. So what happens once one of these action potentials reaches these synaptic terminals? It gets transferred into chemical signals called neurotransmitters, and they go across to the other neurons, where they in turn can excite the neuron to cause an action potential. So the strength in which these neurons communicate, these synapses, isn't static, and it's plastic, and it's determined by the neurons themselves. We say cells that fire together, wire together, and this process of synapses strengthening or weakening is called synaptic plasticity. So what is crucial for neurons to communicate effectively is that their internal ion homeostasis remains intact. So they need to be able to discharge these electrical signals, which involves both negative and positive ions. But in healthy neurons, it's really critical that there's a balance. Glutamate is the most abundant neurotransmitter that's used to convey these electrical signals. So in the synapse that I showed earlier, you have these vesicles that contained neurotransmitter glutamate. When the action potentials reaches the terminals, these get released and these glutamate bind to glutamate receptors on the other neuron. This in turn can open little pores, these glutamate receptors, and positive ions can flow in. And this can, what we call, depolarize the cell enough for it to be able to fire an action potential. Now, the tricky thing is, if there's too much glutamate and you get too many of these positive ions, specifically calcium, flowing into the cell, it can actually cause the cell to degenerate and die. And this is called excitotoxicity. So excessive glutamate can cause hyperexcitability. And this is a real problem. And what's important to know is that this imbalance can be created by things like excessive glutamate, but it can also be because of reduction in chloride channels. So chloride also plays a pivotal role in neurodegeneration caused by hyperexcitability. So hyperexcitability can be caused by excessive excitation or by a reduction in inhibition. So how does this relate to MND? So what we know in MND is that motor neurons that either connect to other neurons or the ones that excite the muscles, they degenerate. So they stop functioning and eventually they will die. And what we find prior to the neurons actually dying and stopping working is that we observe this hyperexcitability. So there's this imbalance between these ions and the ion homeostasis within these neurons that make them hyperexcitable. So what causes MND? What causes these neurons to die? We don't know. We know there are environmental factors, so exposure to chemicals like heavy metals can increase the risk of developing motor neuron disease. So does lifestyle. Joining the army increases the risk of developing motor neuron disease, but neither with these environmental or lifestyle factors is it deterministic. So not everyone who joins the army or is a highly active athlete will develop motor neuron disease. And another important factor is genetics. And actually some cases where people have specific mutations on genes are deterministic. So one of the mutations, I think, one of the genes many of you are probably familiar with is the SOD1 mutation. So certain mutations in these genes, so little mistakes in the DNA, will actually cause someone to develop motor neuron disease. And when we try and find cures for motor neuron disease, we make use of the fact that we know that certain mistakes in the DNA will give you motor neuron disease. And we use this to create mouse models. So there are a few mouse models that people use that have mutations in the SOD1 gene. There are other genetic factors and genes that are involved in motor neuron disease, some of which are deterministic, but a lot of them, there can be some sort of genetic predisposition, but you still need a combination of environment and lifestyle factors to eventually develop motor neuron disease. So really only 10% of people with any individual with motor neuron disease, we can say what the cause is. So in the other 90%, we really don't know what causes MND, and we call these cases sporadic. What we do know is that in all the cases, the genetic cases and the sporadic cases, multiple pathways and factors converge into eventually causing this hyperexcitability. So we see this in all motor neurons, regardless of what the potential causes are. So the drug that's on the PBS for motor neuron disease at the moment is riluzole. It's been the main treatment for motor neuron disease since 1995. And it turns out that this drug dampens excitation. So riluzole is a sodium channel blocker, and what it's believed to do is reduces the amount of glutamate that's released from these synaptic terminals, which means you get less glutamate in the synaptic cleft and you get less glutamate receptor activation, which then causes less of these positive ions, including calcium, to flow into the cell. So riluzole is a neuroprotective drug that blocks glutamatergic neurotransmission, thus reducing excitability. Unfortunately, here's another picture illustrating that if we have this same motor neuron innervating muscles, it gets lots of glutamatergic input from neurons, and that riluzole would reduce this, and therefore reducing the excitability of this example neuron here. What I was going to say is riluzole has severe side effects, and I think anyone familiar with the drug knows this very well. It can cause nausea, diarrhea, and people generally feel really unwell. And it's also far from a cure for MND. Now, this is really not surprising because these sodium channels that it blocks are important for many types of neurons. So riluzole is not motor neuron specific, and taking these drugs will have effects on many processes, unfortunately, not just the motor neurons. Now, going to one of the emergent treatments, which is gene therapy. So with gene therapy, we are now able to replace, repair, or reduce defective gene material. Now, not long ago, gene therapy was really considered kind of science fiction, like artificial intelligence. But in recent years, there have been quite a few strides and progress in both AI and gene therapy. So just to go through what it does. So most of our genetic material is contained in the cell's nucleus, in chromosomes. And in these chromosomes, we have our DNA tightly packed, and bits of that DNA code for specific genes, like the SOD1 gene. So these are the building blocks for proteins. And in the case of SOD1 mutation, what happens? The mistake in the DNA will cause the protein to not function well, and not just not function well, it actually becomes toxic for the cell. So now you have this protein that is making the cell sick. So very, very recently, tofersen, which I think might have had a name change as well, was developed for MND for people that have specific mutations in their SOD1 gene. And this specific form of gene therapy targets the messenger RNA. Now, what cells do is they make copies of their DNA within the nucleus, and they send it out of the nucleus where it gets used to make this protein. And this tofersen basically prevents protein being made from this messenger RNA. Forms of gene therapy where you need the gene delivered directly into the nucleus require a different type of delivery system. Now, viruses are perfect at getting their DNA delivered to the nucleus. So what a virus will do is it will infect the cell, go straight to the nucleus, deposit its DNA, and the cell thinks, oh, we need this protein, but really it's just creating new virus particles. And what we can do now is we can hijack the virus cargo, take out anything that would cause new viruses, and put our gene of interest there. And very recently in Australia, there's a drug for a hereditary eye disorder approved in 2020, and one for spinal muscular atrophy in 2021. For all the three types of gene therapy that I named here, we know what mistake is made in the gene. So far, what you do is you try and correct this mistake. So you replace, repair, or reduce the defective genetic material. What we're trying to see is, can we use this AAV gene delivery therapy to target hyperexcitability in motor neurons? Because we know that this hyperexcitability in motor neurons is seen in also the sporadic cases in MND where we don't know what the cause is. And even though there's many researchers worldwide trying to pinpoint exactly why an individual gets motor neuron disease, it might, as a community, really take us a while to figure that out. So what we're trying to do is kind of take a shortcut and say, look, we don't know exactly what's happening, but we do know that these neurons display hyperexcitability before they die. So our approach is to try and rebalance excitation using gene therapy. So in the healthy state, there's a balance, then we get the hyperexcitability and we get an imbalance, we get an increase of these positive ions. So we are not going to try and block the amount of sodium and calcium that goes in the cell due to the release of glutamate, which is kind of how riluzole works. Instead, we're targeting the inhibition of the neurons. And it's been shown both in mouse models and in humans that there's actually a reduction in certain types of inhibitory receptors in patients and in mouse models. So we're going to try and increase the amount of chloride that goes into the cell and balancing this back to a healthy state. So to do this, we need a few things. We need a mouse model. So we have a SOD1 based mouse model. We need a silencing receptor packaged in one of those AAV viral vectors. And then we have a drug that activates the channel, which is already on the PBS, but for a different condition. So what happens when we inject the virus into the mice? We get expression of the receptor in the cells. And then with this drug, we get an extra flow of chloride, which are the inhibitory ions in the cell, as a way of counterbalancing the excess excitation that we see in motor neuron disease. So some of the data where we record, because that's one of the things that we can very successfully do in the lab here, the activity of the neurons, we show that when we insert our receptor and we activate it with the drugs, we get an increase in the events, synaptic events that let chloride in. And we can also show that per event, we get more of this negative current, more inhibition flowing into the cell. Okay, so of course we want to know, it theoretically, it all looks good, but does it actually work? So in this SOD mouse model, our first experiment is that we treated the mouse pre-symptomatically, which is of course not feasible when we do this with humans, because people would present with symptoms and then you treat them after. In this case, we started the treatment before, which means we can study disease onset. Does early treatment delay disease onset? Do symptoms appear later in time? And then if we follow these animals, do they survive for longer? The data I'm going to show you is a very preliminary data set that doesn't have enough animals. So even though it looks promising, it's not significant by far. We can't draw any conclusions of this yet. But the black lines are the controls, the green lines are the treated animals. And it seems that indeed, by pre-symptomatically treating these animals, disease onset is delayed and survival is increased. If you would relate this mouse days to humans, we're talking maybe 2 or 3 years. And in this figure, this is just a different way of representing the same data, but this is the age of the mice in weeks. And what is natural is for the mice to gain weight But then when they start showing signs of MND, they will start losing weight. And you can see that it takes a lot longer for the treated group to start losing weight compared to the control group. And also that there's, here there's still treated mice left where there was no control mice that survived this stage. So what's to come? Thanks to the Fat Rabbit Innovator Grant, we can extend these first initial results. And one of the big thing, of course, that we want to do is we want to see whether or not starting treatment after symptoms is actually going to make a difference. So can we increase lifespan after treatment? And we can look at motor strength and coordination as well. And then we just want to have a deeper look at the motor neurons themselves. So this is an example image of a mouse spinal cord section where the motor neurons are labeled in green. So what we also want to do at different stages of treatment, see if we have an increase in survival in the amount of motor neurons that are still there compared to animals that were not treated. That brings me to the end. I'd like to thank everyone here. It really was a pleasure to share some of our research with you. And I'm incredibly grateful to everyone behind the Fat Rabbit Innovator Grant, especially Lisa, everyone involved in fundraising, the people from MND Australia, MND and Me Foundation, and really Pankaj and Joe, who really got the ball rolling on this project. Our lab manager and Lee, who makes all the virus, and QBI is a fantastic place with some incredible facilities. This type of research really involves lots and lots of people. We have great animal house technicians and the microscopy facility, histology, and then all the supportive people from the MND UQ Collective, which is the group of researchers here at UQ that come together occasionally to just troubleshoot and talk about our research. Any questions? I think you can put them in the chat. You can also send me an email. And that brings me to hand over to the next speaker, which is Professor Woodruff. Thank you, everyone.
Thanks, Margreet. Hi, everyone. I'll just share my slides. All right, well, thanks again to MNDRA for this opportunity to present some of our work to you on this immune system that we call complement. And you can see that today I'll be presenting to you about the therapeutic targeting of this immune system for the treatment of motor neuron disease. So I have to apologize to people who may not be familiar with the immune system in great detail, but I hope to take you through some of the basics of this particular immune system that we call complement. It's actually an immune system that was the very first evolved. It's the OG, as we say. It was about 1 billion years old in evolutionary terms. So it's in the majority of animals on this planet. But despite being so old, it was really discovered after many of the other immune systems had been already discovered. So it was discovered about 100 years ago by Jules Bordeaux, who in part for this work won the Nobel Prize. And so this is a system that is present in our bloodstream and also many cells in the body, and it's responding to pathogens. It's an immune system. So when the body is invaded by foreign pathogens, but also things like cell injury and protein aggregates that might be relevant to motor neuron disease, this system becomes activated. And I'll take you through some of the consequences of this immune system activation in the next few slides. But we know that one of the major components of this system is a factor called C5a, which binds to its receptor, called C5a receptors, and this drives an inflammatory response. And so whenever you have an injury or a cut to your arm and you see that reddening and swelling that you get and a little bit of pain, you know that your complement system is at play, driving many of these inflammatory mediators that lead to that pain and swelling. And really the ultimate goal of this system is to recruit immune cells to the site of where activation occurs, and then to drive an immune response to resolve the injury. But in recent years, it's been shown that this system can be turned on in many diseases and drive many of the consequences of these diseases. And we know in the brain, for example, that this system is particularly relevant in the context of immune system activation in the brain. And so I'm not expecting anyone to really take anything from this slide other than to understand that the system is quite complex. I myself have been studying this for 20 years and I still get confused sometimes when I see these diagrams. But for anyone who is interested in knowing a little bit more about this particular, this immune system that we have, I highly recommend visiting this particular YouTube channel, or this YouTube video called Tiny Bombs in Your Blood, which I think does a fantastic job of summarizing what the complement system does. And just a couple of quotes that I pulled out from the video itself. You can see here there's a lot we still don't know about this complement system. And in fact, the complement system can kind of be thought of as the central mediator of many other immune systems. So many other systems are really there to activate the complement cascade. And it is a little bit complicated and overwhelming, as they say in the video. So I've really simplified it for you on this particular slide. It's in a cascade, so you can see that each factor subsequently gets activated by the upstream factor, and this allows for an amplification process to occur. Just to confuse, I guess, many undergraduate student, I find is that the proteins were named in the order they were discovered. So you actually get C2 jumping to C4 and then back to C3, again, just to confuse everyone. But ultimately, at the end of the cascade, you get formation of this complex called the membrane attack complex. And as the name suggests, what it does is it attacks cell membranes. And so where you get activation of this system, you actually end up killing cells through a process called cell lysis. And it's been shown that this pathway is potentially involved in the killing of motor neurons in motor neuron disease. Another important process that occurs following activation is this process called opsonization. That is where proteins and cells can get tagged by these molecules. And I've already introduced this cascade called inflammation, which drives many immune processes in the body as well. And so really, to just jump through this, we know that there's opsonization that can be involved in motor neuron disease by tagging these aberrant proteins like TDP43 and SOD1 that can accumulate in patients with MND. We generate these pro-inflammatory mediators that drive the inflammatory response. And we also can directly kill cells themselves through this process of cell lysis. And so complement has been studied for many years in many diseases because it has such central roles in driving inflammatory and immune responses. And way back in 2007, there was the first approved drug called, which was an antibody that blocks C5, one of these central components. And then there was a really long gap until the next complement inhibitors came along. And now there's really a wave or a flood of these molecules that are coming out in the market in recent years for various diseases. But these approved drugs are really to target peripheral inflammatory diseases of the kidneys, the lungs. In the case of COVID, it's an approved COVID treatment for one of these drugs, and also some neuromuscular diseases as well. And if you look at where the drugs are currently targeted, they target at the early parts of the cascade, at the middle parts of this cascade, and this is where they're currently targeting in the cascade itself. So what about motor neuron disease? So if you look back in the literature, back in the 80s, there was lots of evidence that complement could be involved in motor neuron disease. And this was by taking CSF samples or plasma, but also postmortem tissues, motor cortex or brain and spinal cord from patients who've succumbed to ALS or MND, and staining for complement factors. And it was shown throughout all of these studies that complement proteins are upregulated, they're increased in patients with MND. And this is suggestive that this immune system is switched on in this disease and may be contributing to disease processes. And some more recent studies using a bit fancy techniques that I don't need to go into, but this is a recent study where they basically looked at all of the genes that were expressed in motor neuron disease spinal cords, and compared them to healthy individuals. And you can see really the colors, anything that's colored that's not black is showing an upregulation. And you can see in all of these complement genes are upregulated in these patient samples. So we know that complement is switched on or activated in patients with MND. And so we've done a lot of work using this particular mouse called the SOD1 transgenic mouse that Margreet introduced. And it's basically a mouse model that has symptoms similar to what occurs in human motor neuron disease. And these mice progress through various stages of the disease and eventually succumb to the disease. And so we can use this as a model of looking for complement activation in this particular mouse model. And what we find is if we take a spinal cord tissue from a healthy mouse, what we call a wild type mouse, and we stain for complement in the spinal cord tissue, we don't see any complement. That's normal. You shouldn't really see much complement or inflammation occurring in these healthy spinal cord tissues from these animals. So the red basically is showing there's no staining. And I don't think you need a PhD to understand that on the right, we see a massive upregulation of complement occurring in these so-called SOD1 MND mice. So this is at the end stage of disease where we take these animals and we have a look at their spinal cords, and you can see as opposed to the healthy animal, there's quite clearly a strong activation of complement occurring in these mice. And so taking that knowledge to the next steps, what we were really interested in doing is seeing if we could inhibit complement or take out the effects of complement in these MND mice, these SOD transgenic mice, and see what effect that would have on their disease. And so what we started to do is we started to backcross or breed these SOD transgenic mice with other mice that have a genetic deficiency in many of the complement genes. And I'm showing you here one of the key inflammatory genes that we study called C5a receptor. And when we compared the behavior and also the survival of these mice to each other, what we found was that the mice that didn't have any C5a receptor signaling or C5a receptors in their system, these MND mice had an extended survival compared to mice that did have C5a receptors, the normal SOD1 mice. So the genetic absence of these complement genes significantly increased the survival of these mice and slowed their motor neuron death. And we've done a lot of work now looking further at the mechanisms of how this may be occurring. And we certainly see a reduction in these complement activation within the spinal cords of these MND mice, as well as reductions in inflammatory mediators as well. And so the next step of our research was to take this to a therapeutic treatment. And so many years back, we had worked with this particular class of compounds that are small inhibitors of the C5a receptor. This particular drug here is called PMX-205. And we know that this drug is a very effective inhibitor of these particular complement receptors in mice, but as well as in human cells. And so we're able to take this drug and treat mice with this compound. And so we could look at mice, SOD1 MND mice that had drug, were consuming the drug. We were giving this in the drinking water versus mice that were not receiving drug. They were just receiving normal drinking water. And when we did this experiment, we found very similar to what we found with the genetic absence of C5a receptors, that again, we saw an extension in their survival. So mice that were taking this drug in the drinking water had an extension in their survival. They also had reduced motor neuron loss and improved muscle performance compared to the untreated animals. And I think a graph is useful to show you the data that we obtained in this model, but it's also interesting or nice to see an actual video of these mice. And so here is an example of one of these SOD1 mice. It's around about what we call the mid-stage disease. So it's still able to walk around the cage, but hopefully you can see that this mouse is a bit shaky on its hind legs. It's kind of wobbly as it moves around the cage, and it's unable to right itself on its hind legs because its muscles are quite weak. And on the right here, what we have is a mouse that was, I'm sorry, I'll go back. I'll try that again. On the right, we have a mouse that was treated with this drug called PMX-205, and hopefully it's quite evident that this mouse is a bit more mobile. It looks a bit healthy. These mice were actually littermates, so they had the same parent, so they're effectively identical twins of each other. And hopefully you can see that difference between this mouse that's received drug compare to the mouse that doesn't. In fact, you can see just before it was able to right itself, it was able to rear on its hind limbs, so it's got much more muscle strength compared to its twin brother on the right, on the left. Okay, so the combination, I guess, of the work that we had performed over the years, as well as the work of many others, had really pointed to the fact that complement might be an interesting target to potentially therapeutically treat patients with MND with some of these inhibitors. And I mentioned before that there were many approved drugs now on the market for other diseases. And so in recent years, there's been a number of clinical trials undertaken using some of these treatments, these approved treatments for other diseases in patients with MND. And so I'm going to take you through 3 of these trials that have now been completed and take you through some of their results. So the first of these is called the Champion ALS trial. It was an efficacy study looking at this particular drug called ravulizumab, which is a C5 blocking antibody. It was actually one of the very first complement inhibitors that was on the market for some rare blood disorders. And so in this study was a phase 3 double-blinded, randomized and placebo-controlled trial. It was run in the US in particular, but also in some sites across Europe and Asia, and it recruited 382 sporadic and familial ALS cohorts. And you can see here some of the parameters that they were using in this trial. But unfortunately, the review of the data at this interim analysis by an independent monitoring committee did not observe any efficacy in the trial, and so the trial was discontinued. We haven't yet seen the data from this trial. This is just a recent update provided to the public that they have discontinued this particular trial. Another trial that has also completed is this Healy platform trial, which some of you may be familiar with. This was using a compound called zilucoplan, which is another C5 inhibitory protein, not an antibody, but a very large protein that blocks C5 in a similar mechanism to the other compound I mentioned before. And this Healy trial is a ongoing study in the US that is looking at multiple treatments in a patient cohort, and they do various randomization and crossover protocols to enable more rapid throughput of drug treatment tests in patients with MND. And so this particular drug was put through this trial, and similar to what I showed you before, an independent committee looking at the data at this interim analysis point also found that there was no significant benefit in patients taking zilucoplan versus the placebo. And so again, the study was halted at this point and did not progress further in the study. The final trial I want to mention is this particular trial that, again, has just finished. It's called the Meridian trial, looking at this particular drug called pegcetacoplan. Not a great drug name to try to pronounce. And it's actually different to the other 2 inhibitors that I mentioned before, and that this is actually a C3 inhibitor. So it acts at a different site in the cascade to the other molecules I mentioned. And this study was looking at 228 subjects that were enrolled and randomized 2 to 1. So we ended up having 152 patients taking these subcutaneous injections twice weekly compared to placebo. And this is some of the inclusion criteria and exclusion criteria, as you can see here. Again, looking at efficacy in the treatment period on patients with drug. And again, similar to the other 2 trials, this outcome of this particular study was also didn't reach its primary endpoints or its secondary endpoints by looking at various parameters as listed here. And based on these results, the companies have discontinued development of this particular drug, the systemic delivery of this drug for ALS. So the results of those trials are quite disappointing, I guess, given a lot of the hope that was around at the time for blocking complement, this immune system And I want to take you through, I guess, my thoughts on some of the reasons why these trials may have particularly not reached their primary endpoints. And so I guess the one thing to point out is that there's a lot of drugs that have gone into MND and ALS trials, as again you're probably aware, and many of these do fail to reach efficacy readouts when they go to patient trials. And this is probably because MND is quite a complex disease. There's a lot we still don't know about the causes and the contributors of this disease process. And as I said, many of these drug targets have been difficult to establish efficacy when they go to clinical trials. And the other point to note is that even though both 3 trials have been discontinued, there's been no results being released from any of these studies. And so it's really hard to make any informed decision about the reasons why they may not have progressed in the trial, because we haven't really seen any of the data. And so really the question is all hope lost for targeting complement in MND? And hopefully on the next few slides, I'll show you that I certainly strongly believe that there certainly is still hope for targeting this cascade, this immune system in MND. And I think the first really important point to understand is that these drug trials were using compounds that had been developed specifically for peripheral diseases, so blood diseases, kidney disease, lung disease, so these are and muscle diseases. And so diseases where these drugs can be administered systemically, so they're administered either intravenously as an injection into the bloodstream or subcutaneously, which is an injection just under the skin. And the idea of these compounds is that they are not necessarily thought to enter the spinal cord or the brain. And really, at the time, these were the only available complement inhibitors available to test in patients. And so I think the companies were conducting the trials on the hope that perhaps that they might be enough. But importantly, we know that the drugs do not cross the blood-brain barrier, and so they cannot target complement within the brain or spinal cord of MND patients. And if you have a look at the classes of complement drugs that are in development or approved, really don't worry too much about this slide, but just to understand that there is a whole range of therapeutic entities that can be used to target complement. And when it came to the trials, it was really focused on these 2 classes here, these antibodies and also these large peptide molecules. And I think it's pretty well established in the field that you really do need small molecule approaches or even gene therapy approaches, as Margreet introduced, in order to get inhibition of targets within the central nervous system, within the brain and spinal cord, that would be necessary in MND. And so I think this is one reason why potentially these particular trials may have failed. And so we know that complement is expressed quite highly within that spinal cord and within the brain of MND mice, as well as humans. And so you might remember that slide I showed you before of that large complement red expression as shown here. And this was really showing you that complement is highly expressed in the brains of MND mice as well as humans. We've also treated mice with a C5 inhibitor, and we did not see any improvements in survival in this mouse model. And that's very different to what we see when we actually inhibit the C5a receptor genetically, or we treat them with what we have as a brain penetrant C5a receptor inhibitor. And so I really believe that targeting complement is important to do so within the brain and spinal cord. And some of the next generation complement drugs, some of which we're working on, we believe may be better at having potential success in MND clinical trials in the future. And this is just that data I mentioned before. I've shown you this data on the right already. This is our drug PMX-205 that does extend survival. But when we used a C5 inhibitor, that's a very large molecule, at least 10 times the size, we didn't see any extension in survival. And we know this drug is unable to cross into the brain, cross that blood-brain barrier. So just finally to introduce a current clinical study that is ongoing is by a company called Alcinex that has orphan drug approval for PMX-205 for MND. They've completed some of the early clinical development of this molecule and also a safety trial in healthy volunteers. A phase 1A study has been completed. It's shown to be safe. And they're currently conducting a phase 1B study at the Wesley Hospital in Brisbane in a small cohort of MND patients, dosing them with PMX205, and importantly, trying to assess whether the drug is able to get into the brain by collecting CSF as well as blood from these patients. And this will be really useful to understand whether this potential therapy may be a better way to inhibit complement within the spinal cord of patients with MND. And so just to summarize, we know that complement is activated within the brain and spinal cord of patients with MND as well as animal models, and that this potentially drives this inflammatory process that can lead to motor neuron death and disease progression. However, targeting complement within the peripheral blood system is not effective in MND trials, and it's an unfortunate result of what's come out from those 3 clinical studies, but we think that the inhibition of complement with brain penetrant drugs might be more effective. We know they're more effective in models, and we hope that they're more effective in patients with MND. So targeting within the brain may be needed to slow progression of disease in patients in the future. And so I'd just like to thank my team at UQ. We work right next door to QBI in the School of Biomedical Sciences, a very talented group of early career researchers that have really built up a lot of this work in the motor neuron disease field. I'd like to thank our clinical collaborators. We've worked with the Royal Brisbane Hospital, Pam McComb and Rob Henderson, as well as Alcinex. And finally, really want to put a great shout out to MND Australia, who've supported our work over at least a decade now or more, and have really set us on the path to trying to discover new treatments for MND, as well as some of the other funders involved in this work. And so thank you, and happy to take questions.
Great, thank you, Trent and Margreet, and sorry for my very poor pronunciation of your name, Margreet. I've got no way of trying to give it the Dutch treatment. And look, I really appreciate it, Trent. I was a bit disappointed by all those results in the complement system. And it is good to see a very clear explanation of perhaps why they didn't work and why we should still have some faith in complement going forward. And very much appreciate you putting up some unpublished data as well, Margreet. It's great you don't see that very often these days, so it's great for people to put it up. So it's a bit of a question for both of you here, is that you've both sort of put up therapies that can really target what appear to be quite key systems in MND, such as excitotoxicity or neuroinflammation, and even in the mouse models where you've got good effectiveness, but we're still not shutting the disease down totally. It's still, you're delaying the disease, or why is that? Why can't we just, why can't we just shut it down?
Margreet, did you want to go first?
I'll let you go first.
You'll let me go first, okay. Look, it's a great question, and I think it's important to note that in some diseases you do need to have multi-targeted therapies in order to get the best clinical success that you can. And my view has always been that, and you see this a lot in those big reviews on motor neuron disease that come out, that you have about 5 or 10 or so kind of arrows all pointing into the disease process itself. One of them being, as you mentioned, neuroinflammation, and another one being excitotoxicity, for example. And it may be that we do need to have multiple approaches targeting multiple pathways in order to have the best success at beating this disease. In saying that, I think there's still, hope and potential that by targeting one of these pathways, you might still get quite a substantial benefit in patient outcomes. Although it might not be a cure, it might be something like doubling lifespan, for example, or having a significant delay in progression of their muscle weakness. And this could have profound outcomes for the patients themselves and the families. So I don't think all hope is lost by just targeting one of the aspects. But I think you're right. It's important to understand that these treatments that we're looking at are not necessarily cures for the disease, There's certainly meant to be more of a symptomatic, or sorry, not a disease-modifying treatment that slows the progression of the disease.
Yeah, that's exactly it. And I mean, that is a shame, but, you know, I think all neuroscientists will tell you, like, the brain and the spinal cord, it's complicated. When it comes to like an organ like your heart, we know it has 4 chambers, you know, we've kind of figured out how the system works. But when it comes to the brain and neurons, there's just still a lot that we don't understand. And, you know, that basically prevents us from basically getting an easy way in to target the problem. So lots of research is needed, which, you know, is unfortunately the way it is. Like, the brain's an interesting, fascinating place, and it's, you know, really nice to try and work and figure things out, but there's still a lot that needs to be done. But, you know, like I talked about gene therapy and technologies, just the amount of tools and new ways of doing research has really exponentially increased. We're able to do things that 20 years ago we wouldn't consider we could do. So, you know, we'll get there, but unfortunately, maybe not anytime soon.
Thank you. And a question for you, Margreet. You were talking about using the AAV to deliver to increase the chloride transport. So I might have missed this, but is this specific to motor neurons, the delivery? How come we don't get a sort of suppression of the whole excitotoxicity system, or not excitotoxicity, the whole signaling synapse signaling system if you've regulated chlorine transport across all the synapses? Do you run the risk of really damaging the nervous system if it's not specific?
Yeah, so what we can do is with these AAVs, they're different serotypes. And for instance, we can develop a type that gets taken up specifically by motor neurons that synapse on muscle. So if we would do intermuscular injections, this gene would only be exported to the motor neurons that synapse on those muscles. And also, yeah, then they would be the ones that would get this increase in chloride when those receptors are activated. So with these viruses, we can make them cell target specific.
Okay, cool. And have AAVs been used widely in other diseases? Are they a sort of well-tested drug delivery device in humans?
So there's still a lot of research going on about that. So there's 2 approved by the TGA. One is for the hereditary eye disorder, and one is for spinal muscular atrophy. So currently there are 2 being used here in Australia, but only very recently. Like the first one was only approved in 2020. So it's really something that's emerging, but it has really grown in a really short amount of time. And it may turn out that these AAV viral vectors aren't the best, and people are working on other types of vectors and delivery methods as well. So in 10 years from now, this field could have also changed a lot. So for our research specifically, the AAV viral vector might not end up being the one that would go to clinical trial. But if we can show that targeting hyperexcitability by increasing chloride, we might be able to look at other ways as well. And one of the things that we do, that certain receptors have slight variations. So even if we can make drugs that wouldn't target, like riluzole, target all neurons, but just a specific subset of them, because motor neurons, they are slightly different. Like all the different types of neurons, they can be distinguished by small markers. And lots of people are researching around the world as well to just get a more detailed idea of what makes a specific cell that specific cell. But it's just, you know, it's a lot of work, but we're getting there.
And I guess is it one advantage of AAVs? Do they last longer than currently? I know with say tofersen, it's a monthly intrathecal injection, which is obviously quite invasive. So with AAVs, can you sort of get a much less frequent dosing regime?
Yes, so what the AAV does, because it targets the gene to the nucleus, it stays there. And the advantage of the AAV, it doesn't get inserted into the genome, because some viruses, when they basically just latch on into a specific spot, they can actually cause problems, because if they insert in the middle of a gene, that's a problem. But the AAVs actually put the gene into the nucleus, but it doesn't mess with the chromosomes that are already there. And it basically stays there. And so when I do the injections in mice, it only requires a single injection, and they will basically express the receptor throughout their life.
So I think you did mention it on one of your slides, Trent. Are there AAV approaches in complement at the moment being developed?
Yeah, I think there's a few genetic approaches, one of them being AAVs. There's a number of different mechanisms and methods that pharmaceutical companies use to target the genes. And yes, I think AAV is one of them. They're very early stage though. I think at the moment they're being used for eye treatment for eye disease like macular degeneration, because you get a much kind of site-specific effect, not so much for brain disease. But you're right, this could be an approach to think about in the future to enable delivery to the CNS, to the brain, and have that effect on complement within the brain. That could certainly be an approach.
And I think you were saying that one of the reasons complement is sort of perhaps involved in MND is due to its role in helping cells overcome protein aggregation. Has it been used in other diseases where protein aggregation is a key factor, say Alzheimer's, or I think, is it Parkinson's? They get some protein aggregation as well. Have they tested the same drugs in those diseases, or are there other drugs being developed that could cross over into MND?
Yeah, great question. I mean, you're right. These other brain diseases that involve accumulation of protein aggregates and progressive loss of neurons in the disease, like Alzheimer's, Parkinson's. Yeah, complement's certainly been shown to be involved in those diseases. Again, from a same level of evidence that we have for MND, we know in the mouse models it's activated. We can block it in the mouse models and slow progression of disease. And in humans, at least we see upregulation of these proteins in the brain in patients. There's been no trials though for these diseases, at least as far as I'm aware. And I think those diseases are much more costly in some ways to begin. And but I wouldn't be surprised if in the next few years there's also attempts to try to target these particular diseases as well. I think one of the advantages that the companies thought for targeting MND was that there is a neuromuscular involvement obviously in this disease as well, and perhaps it was thought that targeting, even though they're peripheral restricted drugs, that targeting that activation within the muscle, the neuromuscular junction itself, may have some benefit. But yeah, like I said, from our data, it doesn't seem like inhibiting complement in the periphery has any effect in the mouse model. So I'm not surprised that there was minimal, I guess, efficacy in the clinical studies as well.
And then just a final question to finish up with this. So this is coming from our audience. I guess for both of you and for all of us, possibly, is it finding it how frustrating it is when we see these trials being stopped and then we don't see the data being published for a long time? I mean, it's not just these recent ones, but I don't know if you want to comment about that apart from gnashing your teeth a bit.
Yeah, absolutely. I mean, I guess, we have to give a bit of, fairness to the companies themselves. They've invested a lot of money in developing the drugs, and there's obviously proprietary, secrets in some ways that they want to hold on to. But absolutely, seeing the actual data from these trials could be very informative for for future research and directions. And you know, often there's analyses that can be conducted once trial results have been released, and you might find something interesting in that data that you know may not have been seen before. So yes, absolutely, it's very frustrating, and I really do hope that some of this data is released in the future, so we can have a look at it and really try to sink our teeth around and what could have been happening in those trials.
Great. Well, thank you both. It was fantastic to hear some. I mean, I think our last state of play we had talked about genetics and the kind of more about the causes. And it's great to hear about approaches we can actually treat some of the more downstream effects in this disease. So thank you both, Trent and Margreet, and also explaining some incredibly complex concepts as well in some relatively simplistic terms. So to everyone else, thank you for watching tonight. Everyone who dialed in, registered, receives a link to the recording. And so feel free to distribute that across your networks and watch it again if you want to brush up on some of the details you might have missed. And to access previous versions, you can find our state of play, all our videos on our website and on our YouTube. So again, thank you for everyone for dialing in. Thank you to Trent and Margreet, and good night, everyone.
Thanks all.